telling you about some rather preliminary results that I've been getting when trying to understand why the species of Phyllobates frogs are what they are and where they are. And so, in our quest to understand why species are what they are, a huge question that biologists have asked even from pre-Darwinian times is where species were they are. This usually explains a lot of processes regarding diversification, because species will either not be able to move places or be able to move places or come back together, as we've been hearing in most talks. And one of the questions about where species are that interests me a lot is the so-called leapfrog distributions, where you have disjunct populations or species that are phenotypically similar, like these two red globes, separated by areas where they don't exist, but other closely related populations with different phenotypes exist. And a rather simple question that can be very, very informative to ask regarding leapfrog distributions is whether the phenotypically similar groups are more closely related between themselves or not. And this can help us gain insight on whether what has happened is actually separate evolution of the same traits, or just clades on their own colonizing or moving in different areas, generating these patterns. And I've been studying this in Phyllobates frogs, which are famous for being so toxic that native Latin Americans actually still use them to hunt in some places. You've become aware of people that are still using the frogs to poison their darts. And a neat thing about this system is that they are phenotypically tractable. There's 2 coloration patterns in currently 5, but as you will see, more species. There are 2 species, and this failed. There are 2 species that have plain yellow colorations, and another 3 species that are dark with a bright dorsolateral stripe running down their backs. They are distributed in the biogeographic Choco forests in Central America and Colombia. They only make it up to southern Colombia and not down to the rest of the Choco. And none of the species are currently in contact. They're all allopatric. The 2 Costa Rican ones and Panamanian ones are divided by the Talamanca Mountains. In Colombia, the barriers are less evident, but they are still separate, and you will never find them together. And what I'm asking here is, of course, whether the 2 yellow species are sisters of each other, or is this the case? And that would help us explain why there are these 2 patches of large yellow species that are also a lot more toxic. And the answer seemed clear back in the 80s when people were doing allozymes. All the frogs had the same allozymes, so the, I guess, natural phylogenetic hypothesis was the 2 yellow ones are sisters of each other. They are more derived in being larger and more colorful, and as I will show you later, they are also developmentally rather similar, so people pretty much had this story finished. They studied these frogs more for the properties of their toxins rather than their phylogeny. But then in 2006, when people started building the poison frog tree of life, now there were molecular methods that people could use. This was awesome. The 5 samples of Phyllobates that were included in that study started telling a different story. So for the past, I don't know, 8 years, the co-authors in this study, and in the later 3-4 years myself, have been sampling these frogs mostly all around their ranges. They had been largely unsampled, so it has been quite an effort. We still have a big sampling gap here, which is where most of the cocaine in the US comes from, so sorry about that. And our approach has been basically genotyping every sample we can get our hands on for mitochondrial DNA, and then we have genotyped a subset of samples for a bunch of SNPs across the genome, like everyone does right now. However, frogs are pretty complicated. You can just go sequence every genome and find, like you would do with humans or with some small genomed insect. Frog genomes are huge and also full of repetitive elements, so I'm not going to go down the alley of actually trying to assemble one of these genomes. So to surpass this, we've been using an exon capture protocol that is very similar to UCE's, but with your own target. So what we did was take a juvenile Phyllobates bicolor, cut it into pieces, and sequence the transcriptomes of 4 different tissues. I didn't put the brain there, because I forgot. Then we split all the transcripts we could find into exons and designed some baits that we then used to enrich regular Illumina libraries and sequence them, and this gave us about 38,000 exons, which we could use to capture DNA. But then we didn't have a genome to map this against, so we just took all the reads from the Colombian species, which, as I will show you, are all very closely related, made our pseudo reference, if you want to call it that. It's just something against which to map, and kept only pieces of DNA that we could actually map back to our targets. It's amazing how much crap you actually pick up when you're supposed to be enriching for the exons. Then called genotypes, as you would usually do, and this gave us about 200,000 SNPs at low coverage for 60 frogs, 62% overall. At about 100 bucks per sample, it's quite a deal, I think. And so now some results. The mitochondrial DNA shows a mess, basically. If you remember, we had the, what's the point? It's very faint. But we, as you can see in the tree right there, none of the yellow clades are sister to each other, and there is a third yellow clade popping up, which are the southern distribution of Phyllobates bicolor. And accordingly, what we thought was one species, which is all these dots here, except for the red and the blue, which were considered Phyllobates or Artenia, just each one formed their own clade. And on top of everything, we had purchased a couple of frogs from the pet trade to do other stuff for my dissertation, and they formed their own little clade here. So it seems like I might have found an unknown species of poison frog in the US. You can buy it from a pet store. So basically, this is starting to tell us, well, that the leapfrog pattern is not because the yellow species just colonized on their own, like down the mountains. And it's also telling us that we were seriously underestimating the diversity in these genes. Now, when you look at the exon capture, this is just a RAXML tree that finished running yesterday, so sorry for the preliminariness. You get almost the same tree, except for 2 important discordances, especially this clade of Phyllobates or Artenia, which are these points, is actually split in half. These 3 points form one clade, and these 2 actually get joined with all of these. This mitochondrial discordance might have to do with the geographic proximity. When we look at a PCA of the SNPs, it tells pretty much the same story. It supports the clades really nicely. So I think the new clades that we are finding seem to be a real thing. And to actually get an idea of the more phylogeographic side of this, we need to flip the map so that you can see the mountains. And this is basically the tree that we're getting, so we're actually finding some more, or getting a better idea of the actual barriers acting here. Baudot Serrania, which is here, seems to be a recent barrier for these populations, which were apparently the first to have diversified. And then the San Juan River, which runs down this valley, seems to be the largest barrier here, which is separating these 2 clades of yellow and striped, and yellow and striped but more messy. And this again tells us that the frogs seem to be writing the geography rather than just being grouped by phenotype. Now, if you notice the PCA, there is quite a mess there of points which correspond to these three morphs, and that is an interesting mess when you actually look at how the frogs look. So I've expanded the map a little bit. You have Phyllobates or Artemia, dark and striped, as I just told you, two different lineages that are currently called Phyllobates bicolor, and then there is this set of frogs that look rather interesting. They have some yellow blotches down their back. They are also incredibly variable. These are all frogs from the same little patch of forest about the size of this room. And as you can see, there is a lot of variation in the amount of yellow dorsal coloration in these frogs, which is very interesting if you look at how yellow coloration develops in all the plain yellow plates, which is that they first acquire the dorsolateral stripes like all the other ones. This is why they were considered sisters, because they had lost this ancestral state that was still present during ontogeny. And I just think this is strikingly similar to the adults of this population I'm telling you about. So where I'm going, and this is where the very preliminary stuff really starts, is asking whether this might have to do with gene flow. So if we look at the Triana PCA, only of the samples from this area that I'm talking to you about, you can see that there is an interesting discordance. Well, first they do separate in different groups, so maybe the big mess from the previous PCA plot was due to just the close relatedness in comparison to the other species. But then the white-banded or the more yellow population of Phyllobates or Artemia actually clusters with bicolor in the tree and with the other Artemia in the PCA plot. So again, thinking about gene flow, I ran some tree mix analyses. I'm also running other stuff which unfortunately wasn't done today. And if you give it 0 migration events, then tree mix will infer the same tree that I just showed you. If you give it anywhere from 1 to 11 migration events, it will only give you a migration event from Phyllobates or Artemia to the white-banded population, which is still sister to bicolor. So this is suggesting that there might be gene flow involved. However, it could be also two differences in effective population size and all the caveats that these methods have. This seems to explain 100% of the variation in the data, so I am quite suspicious of this result, considering it fits amazingly. But so, this is where the rest of my analyses are. My lab is probably hating me because I'm hogging up half of the server. So, in summary, what I've told you is that none of the yellow species of Phyllobates that exist, plus the extra one that we think we have discovered, are sisters to each other. Coloration seems to vary very widely across the tree. There might be gene flow involved, and also I'm going to have to be doing a lot of taxonomy this summer because there seems to be some species delimitation to be done in this clade. But really we're just still starting to find out what these frogs are about, so stay tuned. With that, I'd like to thank my two advisors during this project at different stages, collaborators, and all the institutions that have provided funding and space, and I'm happy to take any questions.